talk. You're all familiar with Ada Yonat's work, and so I'm not going to tell you much about crystallography or ribosome structure, but rather I'm going to focus on a particular problem, which is a fundamental problem that the ribosome is involved in. And that is, if you notice the first slide that Aaron put up, it was about Watson and Crick and blueprints for the structure of DNA. And if you notice what the citation said, it was for the idea of carrying information, of molecules carrying information and information transfer in molecules. And the point about DNA is that it contains the information that we need to make protein, and that's done by transcribing the DNA into RNA, and then the RNA is translated into protein. And the first real step in this direction was taken when a molecule was actually predicted. Now, theoretical physicists often predict particles before they're discovered, but that's very rarely the case in biology. But here, in order to understand how you go from a nucleic acid code to an amino acid code, Francis Crick actually predicted that there would be an adapter molecule. He thought of it as maybe a trinucleotide, but when Copeland and Zemechny discovered this molecule, it actually turned out to be a 76 nucleotide piece of RNA, which base pairs with a triplet at one end, base pairing to a triplet codon on the mRNA, and brings along an amino acid that's 75 angstroms away from the codon. So clearly, this doesn't happen on a naked template, as was originally proposed by Crick based on his trinucleotide adapters, because when cell biologists started looking at where proteins were made and where newly made proteins ended up in cells, they found that newly synthesized proteins localized on these blobs on the endoplasmic reticulum. And these blobs, when they were isolated, were all about 250 angstroms in diameter, and all of them consisted of a large and a small subunit. Okay, and these particles originally were called the ribonucleoprotein particles of the microsomal fraction, because they were isolated from microsomes and consisted of both RNA and proteins. But if you wanted to keep saying this every time you wanted to refer to it, it seemed to be rather too much. And so at a meeting in the late 50s, Howard Dinsis, who also discovered that proteins are made from N-C termini, suggested, why don't we just call it the ribosome? And that's the name that has stuck ever since. Now, many of you know about Jim Watson, because of his discovery of the double helical structure of DNA with Francis Crick, based, as we've heard, on data from Rosalind Franklin. And he's also known for a lot of other things, for his frank views on eugenics and gender and race and all sorts of things. So, but not very many people know that his lab was actually involved in some of the first biochemical studies on ribosomes. Okay, and the reason that he isn't very well known is, apart from a few theoretical papers which he published by himself, he didn't put his name on any of his postdocs or students' papers, because his view was inherited from Cambridge, from the tradition of Bragg and Rutherford and so on, that unless you've actually done part of the work, but nevertheless, people in the ribosome field know about the contributions of his lab. And in 2001, after the structures of the atomic structures of the subunits were determined, there's a symposium, and here he is talking about the early days.
The symposium is on the ribosome, the little round object which doesn't contain DNA, but contains RNA. And I started studying it with Alfred Tisset, who was actually here at Harvard in the spring of 1957. And we knew that it was involved in how proteins are made, because the newly made proteins were attached to it. So we thought if we solved the structure of proteins of the ribosome, we would know how proteins are made. And that was a big puzzle, because had to somehow go from DNA to the information, and DNA to the information in protein. And there was this particle which we thought was round and spherical, and hopefully only contained one sort of protein molecule. If it did, then maybe it would not be easy to solve, but it would be solvable. So over the next couple of years, Alfred and I assembled some people. First, a protein chemist who looked at it and said, I'm leaving. And because it wasn't simple. And then a protein chemist actually said, gee, there are these 30 proteins in this little round thing. And so automatically I knew we'd never know its structure.
So that was Jim talking about the early days of ribosomes. But fortunately, people didn't stop working on the ribosome. And after about 40 years of biochemistry, this is sort of the picture we had of the ribosome, which is that the small subunit binds messenger RNA and presents the codon, presents the codon to the anticodon of the RNA, and the amino acid binds at the peptidyl transferase center. Now, to go from this kind of picture to a real detailed mechanistic understanding of the various aspects, including decoding and peptidyl transfer, required a high-resolution structure, and I'm not going to go into that. I'm sure, since this is Israel, you all know about Yonat's pioneering contributions to the crystallography and how that developed. So I'll talk about the decoding part. Now, decoding means that you have to attach the right amino acid, which corresponds to the codon, which ultimately means you have to choose the right tRNA corresponding to a particular codon. And this is the genetic code table. And what you can see is that each triplet codes for an amino acid. But you now also can see that there are some peculiarities, that often the last letter doesn't matter. It still codes for the same amino acid. And those who think about evolution believe that the code originally started off as a 2-letter code, where the third letter never mattered, but then split up, and now you have occasionally split codon boxes. And there are fewer tRNAs in bacteria than there are codons. And when the code emerged, this led Crick, who was a great theorist, to propose another hypothesis, which is that the same tRNA would recognize multiple codons that differed only at the third position. So the idea is that you must have proper Watson-Crick pairing at the first two positions, but you can tolerate certain kinds of mismatches at the third position. And despite many years of hand-waving and so on, people didn't really understand in structural or chemical terms why it should matter only at the third position and not at the first or second. So the problem the ribosome has is the following. You have the ribosome that's synthesizing a protein, and here's the nascent peptide chain emerging through the 50s. And there's a codon on the mRNA waiting for the tRNA to bring in the next amino acid. But in the cell, there's an entire pool of tRNAs, and these tRNAs are bound up with the elongation factor TU, which is a GTPase. And from this pool, the ribosome has to select just the right tRNA that corresponds to the codon. So, how does that happen? Now, another thing to keep in mind is that the selection is very fast. It's 20 residues per second, and it's very accurate. It's better than 1 in 1,000. Now, if you take the best peptide synthesizers made by chemists today, it's nowhere near the speed and accuracy of the ribosome. So although I'm not a very good chemist, the ribosome certainly is a very good chemist. So how does this happen? Well, we think of accuracy often in yes or no terms. We think of molecular recognition as something like a lock and key, as proposed originally by Fisher, that either it fits or it doesn't. But the first person to think of recognition in quantitative terms was Linus Pauling. And he was worried about a problem in translation, as it turns out, which was, how does the translation machinery distinguish between valine and isoleucine? Now this problem has to do with tRNA synthetases, but the general principle remains. And that is, you can think of an enzyme that's designed for valine as being able to exclude isoleucine because the pocket is too small to take that extra methyl group. But you couldn't imagine why something that would accept isoleucine would reject something, would reject valine, because all that's different is an extra methyl group. Otherwise, it's chemically identical, and the free energy, the bonding from the extra methyl group might not be very much, just some extra van der Waals contacts. So why would it be able to exclude valine? And he suggested that there would be a limit to the accuracy of any system which is related to the free energy difference between the correct and the incorrect substrate. Now, that's true if you have a single recognition and nothing else happens. But in fact, if you have an enzymatic reaction, you have substrate binding and it results in a product, then the correct thing to think about is the free energy difference in the transition state of the reaction between the correct and incorrect substrate. Okay? And really, if you want to think about accuracy for an enzymatic reaction, Alan Fursch also suggested that the real criterion is the kcat over kn between the correct substrate and the incorrect substrate. It's going to tell you the ratio of the correct to incorrect product being formed, and that's ultimately what accuracy is about. So how does this have to do with the ribosome? Well, what it has to do with the ribosome is the following. You have, in the ribosome, one peculiarity is you can tolerate a GU mismatch at the third position, but at the first or second position, it's not tolerated. So that doesn't seem to, the free energy difference ought to be the same, so it doesn't seem to make sense. The other thing is that the reduction in base pairing energy between GU and AU is only about 1 to 4 kilojoules per mole. So if you apply the Pauling formula, that would only result in an accuracy of 1 in 5 or 1 in 10. Okay? But the estimated error rate of translation is better than 1 in 1,000, which would correspond to 15 to 20 kilojoules per mole if you applied Pauling's formula. Now, you could understand why something completely different could be rejected and still have that sort of accuracy. But you couldn't understand why a GU to AU difference, which is only about that much, would give rise to that kind of accuracy. So clearly, the accuracy of translation cannot be explained by the free energy differences between the correct and incorrect base pairs. So, what is going on? Well, the first piece of evidence that the ribosome might be involved in the accuracy of decoding came from a classic paper by Julian Davies et al., where they looked at the effect of an antibiotic, streptomycin, which binds to the small ribosomal subunit. And this antibiotic was shown to increase the error rate of ribosome. And this was the first piece of evidence that something that binds to the ribosome can actually change the accuracy of protein synthesis. And from this, they proposed that the ribosome had some sort of decoding site that sensed the accuracy of base pairing between codon and anticodon. And later on, they found that there were mutations in the ribosome itself which caused resistance to streptomycin. And in the absence of streptomycin, these ribosomes were hyperaccurate. So they were better than normal ribosomes in terms of accuracy. They were also slower, of course, but they had higher accuracy. And then there were second site suppressors to streptomycin resistance in other locations, which made the ribosome less accurate. So you had streptomycin, which increased the error rate, resistance mutations that decrease the error rate, and other mutations that increase the error rate. Now, there was no structure of the ribosome. This was all done in the 60s. And so people didn't know where these mutations were, and they thought, well, these mutations might be right around the decoding site where the codon and anticodon bind, and they may be modulating the accuracy by either helping improve the recognition or interfering with the recognition. Okay? You couldn't rule that out. And so people like Manfred Eigen and Otapo said that the ribosome was like an enzyme that sensed the accuracy or the geometry of the substrate, or the shape of the substrate, and that's how they improved the accuracy of translation. Just like an enzyme would distinguish between a substrate and a similar but not identical molecule, like between glucose and fructose, for instance. Now this idea very quickly fell into disfavor, and because while the structure of the ribosome wasn't known, the structure of tRNA was actually determined in the mid-1970s. And so its structure was known, and so it was known that the anticodon was here, that it would bind to the codon. So it was a big surprise when a mutation was discovered here at position 24 in tryptophanol tRNA that allowed it to read through a stop codon. That means it corresponded to a loss in accuracy. So this is called the Hirsch suppressor tRNA. It was discovered by David Hirsch. And subsequently it was found that if you cross-linked 8 and 13, then you could reverse the effect of this mutation. So you could make that tRNA behave normally again. And then Mike Yaros and his colleagues found mutations in this base triple by mutating this A9 to a C, or mutating this base pair, and those also resulted in loss of accuracy. Now all these mutations were far away from the anticodon, and so you couldn't say that they directly looked at the geometry of the substrate. Okay, that whole idea seemed to be called into question. And just as an aside, Aaron Klug, in one of the two tRNA structures, suggested that this base might actually, base pair with 44 in his structure, but might act like a hinge where it could base pair with 45. And he suggested that this could be some sort of hinge point in tRNA. Now, it's true that that region is flexible. I'll come to that towards the end of my talk, but it didn't act in quite the way that Aaron predicted. So he was on the right track, but perhaps not for the right reason. Now, given this, another idea took hold, because the idea that the ribosome recognizes the shape of the substrate didn't seem to be true. And so people proposed a different idea, and that's an idea called kinetic proofreading. In kinetic proofreading, what happens is the tRNA is brought in by elongation factor TU to the ribosome with the growing polypeptide chain, and this binding is reversible. But if it's a correct anticodon base pair, it results in GTP hydrolysis by EFTU, and after GTP hydrolysis, EFTU changes configuration and leaves the ribosome. The tRNA then swings into the peptidyl transfer center, and then peptide bond is formed. Okay? And this point is the following. At this point, the tRNA could either dissociate or go into the peptidyl transfer center. And at this point, it could either come off the ribosome or result in GTP hydrolysis. So you have two steps where you can have dissociation of tRNA, where you have some sort of equilibration, and they're separated by essentially an irreversible step of GTP hydrolysis, because in the cell, GTP hydrolysis is essentially irreversible because of the ratio of GTP to GTP. So the fact is, if you do the kinetic analysis, you could get as much as the product of the two selectivities at these steps. Okay, and so you could get a much higher accuracy than you could get in a one-step selection process. And at the time, it was believed that the free energy from base pairing alone was about 1 in 100. That turns out to be wrong, but people thought that was the number, and so they thought, okay, if we get 1 in 100 at one step and 1 in 100 at another step, you could get as much as a factor of 10,000. Okay, and that would explain the accuracy. So the ribosome is, all the ribosome is doing is providing a platform for all of this stuff to happen, but not actually playing an active role in the selection process. idea, I don't understand why that idea has been in the textbooks for 40 years, because when you actually think about it, it doesn't make any sense at all. Because here's part of the problem. Part of the problem is that GU versus AU is only a factor of 5 to 10. So even with proofreading, it wouldn't be enough. Okay? Even if you allowed it to totally reach thermodynamic equilibrium, which would be essentially infinitely slow at each step, it would still give you only a factor of 25 to 100. However, no one ever seemed to have thought that you could have incorrect combinations. Here's an arginine on a serine codon, arginine tRNA, which has 2 GC pairs and a GU pair. This is actually should be more stable than a correct combination with 2 AU pairs and a GU pair. The only problem is here the GU is at the first position rather than at the wobble position. So if you were simply counting on the dissociation rates, it just wouldn't work. This would actually be more stable than the correct tRNA, so it wouldn't matter whether you had selected it twice or once, okay? And yet the ribosome, we know, has no trouble selecting the correct tRNA and rejecting the incorrect tRNA. The other piece of evidence came from pre-steady state kinetic data, where Marina Rodnina and her colleagues showed that not only are these dissociation rates different, and they're not that different for correct and incorrect tRNA as we might expect, but she said that she found that the biggest change was that forward rates for the activation of GTP hydrolysis was very different for the correct tRNA. It was much faster for the correct tRNA than for the incorrect tRNA. So what she proposed was that somehow the binding of the correct tRNA is inducing conformational changes in the ribosome that leads to GTP hydrolysis. And so GTP hydrolysis is much faster when the right tRNA binds. So when we solved the structure of the 30th subunit in the year 2000, one of the first things we did was to ask what's going on in the decoding section. And so what we did was to soak in oligonucleotides corresponding to codon and anticodon of tRNA and ask, how does the right 30th subunit look when the codon and anticodon are bound compared to when they're not bound? So here's the codon, so the decoding center is right here, and it's surrounded by 3 conserved nucleotides, which I've highlighted in red. And when tRNA binds, the first thing that happens is, the first thing we noticed was that when the tRNA binds, you have this global domain changes. So you have a closure of the domains of the ribosome around the tRNA substrate. Often this is, many enzymes close in around their substrate. This is a classic case of induced fit. And that domain closure is caused by a local change, where these 3 nucleotides change conformation and bind the minor groove between the codon and anticodon and stabilize the codon-anticodon interaction. It's effectively, you're amplifying the recognition between the codon and anticodon by this interaction of these bases with the minor groove. So if you look at what's happening, what you see is that at the first 2 positions, you see that the ribosomal bases that change conformation are making very tight interactions with the minor groove. But at the wobble position, there isn't any such tight interaction. In fact, what we have here is a GU wobble pair, and you can see the ribosome is perfectly happy with it. And if this were a Watson-Crick pair, since the codon is held tightly, this base would have to move down and there would be a big open space here. So in other words, the ribosome is not looking at the geometry of base pairs at the wobble position, but it's looking closely at the shape of the base pair at the first 2 positions. And now, why is this minor groove interaction so important? Well, in the mid-70s, Siemens and Rich, in their analysis of double-stranded DNA structure, realized that something that was known from Watson and Crick. Watson and Crick realized that the shape of AT base pairs are the same as the shape of CG base pairs. These are called isosteric base pairs. But Siemens and Rich also realized that the N3 of purines and the O2 of pyrimidines are in exactly the same position. So if you flip that base pair around, the O2 will end up where the N3 is, and the N3 will end up where the O2 is. So the idea is that the minor groove not only has the same shape, but it has hydrogen bonding acceptors at the same position. So if you recognize the minor groove, you can check for Watson-Crick pairing geometry, but do it in a completely sequence-independent way. Okay? And that's the point. The ribosome is checking the Watson-Crick geometry at the first two positions and gaining a lot of additional free energy. The free energy that has been measured both by Jennifer Doudna's group and our group, and it's as much as 15 to 20 kilojoules per mole, which would actually be enough to account for the accuracy in a single step if the ribosome chose to be infinitely slow, but we'll see that it doesn't do that. Now, before I leave the minor groove recognition, I want to touch on a general problem, which is that DNA and RNA polymerases also have to ensure Watson-Crick pairing when they either replicate DNA or when DNA is copied into RNA. They have to make sure that you have perfectly complementary base pairs that are added during the synthesis of the new strand. And not surprisingly, DNA and RNA polymerases also very tightly inspect the minor groove, but they do it using conserved amino acids. Okay? What the ribosome shows is you can do that minor groove recognition using entirely RNA. This leads to a bit of speculation, which is, if you imagine that there was a very early RNA world before there were proteins, okay? If you didn't have this minor groove recognition, you would be dependent, when that system replicated, purely on base pairing free energies from accuracy. Now we know that GU versus AU is not that accurate. It's only about a factor of 5 or 10. If you have a very simple template to replicate, okay, maybe you don't mind an error rate of 5 to 10. But if you have a system that's growing in complexity, one that's complex enough to actually evolve a ribosome before proteins could be made, then you must have needed a much higher level of accuracy. And what the ribosome shows is you can do it all with just RNA. So my speculation is that maybe an early RNA polymerase that's now extinct, that's entirely made of RNA, might have evolved minor groove recognition in order to allow a stable, a complex system to replicate in a stable way in the absence of proteins. So why does the minor groove recognition by the ribosome, how does that explain all those mutational data, and why does it lead to GTP hydrolysis? Well, first let's deal with the mutational data. Here's S4 and S5, and here's streptomycin, and here's S12 where the mutations changed accuracy. Mutations here lower accuracy, mutations in S12 increase accuracy, streptomycin lowers accuracy. It doesn't seem to make any sense initially, because these are not right around the codon-anticodon base pair. They're all over the ribosome, all over the 30S. What we notice, though, is that all of these mutations are at an interface between two domains of the 30S subunit. And once we realize this, we look at those conformational changes in the ribosome upon tRNA binding. And what we realize is that those domains move, and there's a domain closure around the tRNA substrate upon tRNA binding. Now, movement of that domains is such that interactions here would be disrupted when tRNA binds, and new interactions would be made here by S12 when tRNA binds. And streptomycin would stabilize the closed form after tRNA binding. So suddenly, a single concept could explain a wide variety of mutational data. And that concept is that you need this domain closure to get to the transition state for GTP hydrolysis for tRNA to be selected. So anything that makes that domain closure energetically more difficult will result in the barrier being higher, and so incorrect tRNAs would have an even harder time activating GTP hydrolysis. So you would increase the accuracy. And anything that makes it easier for this domain closure would make it more likely that incorrect tRNAs could surmount the activation barrier. And that's exactly what's happening. If you mutate this interface, you're going to make it easier to reach the closed form because you don't have to pay the energetic penalty of disrupting those salt bridges. And if you mutate some of these residues which are involved in interactions, you're making it less energetically favorable to get to the closed form. And again, you're going to increase the accuracy rather than lower it. And streptomycin stabilizes something like the closed form and therefore increases the error rate or lowers the accuracy. So all of this suddenly starts to make sense. So then you might ask, why is this domain closure actually required? If that's required for tRNA selection, why is it required? That came when you start looking at how tRNAs are selected. tRNAs are not brought in by themselves by elongation factor TU, which when it binds and when you have that microgroove recognition, results in GTP hydrolysis, and then the tRNA swings into the peptidyl transcript center and results in peptide bond formation. So what we realized is when you look at the EFTU tRNA structure, which was known by cryo-electron microscopy, the movement of the domains that we saw in the 30S would be such as to put that shoulder of the 30S, push it towards the EFTU tRNA complex. So we suggested that the domain closure, by moving towards the EFTU tRNA complex, is stabilizing the transition state, which is, in other words, triggering GTP hydrolysis by EFTU. So, of course, if you want to know how that happens, you really need to get an atomic structure of this complex. And that took a very long time, about 10 years in our lab. So from 2001 to 2006, it took us that long to just get a high-resolution structure of the entire 70S ribosome with mRNA and tRNA. There was a low-resolution structure in Harry Noller's lab, and there was this higher-resolution structure of the empty ribosome from J.D. Cake's lab. But we couldn't get a structure with substrates at high resolution. And so it took us a couple of years to actually make thermos ribosomes that were capable of giving us new crystal forms, and then we had to scale that up to make hundreds of milligrams of ribosomes so that we could try thousands of conditions. And then these two postdocs, Maria Salmer and Christine Dunham, tried to crystallize an EFG complex, which is a factor that's involved in translocating and moving the tRNAs to the ribosome. And when they got crystals, these crystals diffracted initially to 6, and then 4.5, and then eventually to beyond 3. And you know, are you able to see it? So when we got these high-resolution crystals, they were joined by the other people in the lab, and they worked together to build the entire structure, a high-resolution structure of the ribosome with mRNAs and tRNAs back. Now, this crystal form was extremely useful for a variety of things. For instance, we and Harry Noller used this crystal form to understand how release factors recognize stop codons, and then Tom Steitz's lab and Marat Yusupov's lab have also used it for various studies. But there was a problem in terms of looking at EFTU or EFG or any GTPases which bind to the ribosome. And that problem is that in this crystal form, as well as in all the other 70S crystal forms, a protein called L9 sticks out of the ribosome and binds to a neighboring ribosome in the crystal lattice. And it does it in such a way that it occludes the binding of GTPase factors. So even if you have GTPase factors to begin with, you know, this interaction is so strong that in the equilibrium, you only get crystals of the factor-free ribosome. And we did that for many years, okay? And so eventually, when we saw what was happening in the high-resolution crystal form, Maria Salmer and Albert Weizenbaum deleted the gene for L9. And so we produced a strain of thermos that had made ribosomes without this protein L9, that would promote the formation of other crystal closures. And Frank and John actually obtained initial hits of an EFTU tRNA complex, but both of them left the lab. And it was only when Yang-Gui Gao crystallized an EFG complex using that strain that Martin Schneing and Rebecca Voorhees and Kelly put in a lot of effort. Even though they started from these initial hits, they still needed 1,200 trays with 96 drops per tray in order to get 12 crystals that gave us a data set with 3.6 angstroms. And now we're at about 3.1 angstroms of the EFTU tRNA complex. So the question is, what did we learn about it from there? Well, the first thing is that the tRNA binds in a different position in the 50S compared to the 30S. That was already known from footprinting studies of Harry Noller and Danish Moazin. And the other thing we know is that the tRNA is bent, okay? And that was known from cryo-electron microscopy studies. And if you ask where it's bent, you can see that the bend is in the anticodon stem. And if you look at the anticodon stem in detail, you see that the bend is caused by an unwinding or an untwisting of the anticodon stem of the tRNA. And this is exactly where the mutation, one of the mutations that alters fidelity in the tRNA occurs. So clearly the tRNA is distorting there, and if you make it easier to distort, then your energetic penalty for reaching that transition state is lowered. So it's exactly the same idea as the domain closure in the 30th subunit. Anything that allows you to reach that transition state more easily increases the error rate of the ribosome. There's also interactions at the D stem, and if you analyze that, what you see is that, again, there's a movement of about 5 angstroms. And here's where the HER suppressor is, okay? Now this HER suppressor was not very clear. It's in the region of distortion, but this is a G to A mutation. It's a GU pair that mutates to an AU pair. And it wasn't clear why an AU pair should be less stable than a GU pair, and it wasn't clear why the HER suppression should work. I'll deal with that in a minute. But here's why the crosslink works. If you crosslink 8 and 13, then you make it less favorable for it to distort. So if you make it more favorable to distort here and less favorable to distort here, then you cancel out the effects and you get a more or less wild-type phenotype. And this is a region where fluorescent reporters were attached in the kinetic studies, and you can see why its environment would change during the decoding process and why it would be such a good reporter for looking at decoders. And finally, here's the base triple, where if you mutate A9, you destroy this triple, and so you can make it easier again to distort. So again, everything makes sense, except for the HER suppressor, which is a G24A. And here's what happens. You have a GU mutation, but near it is another G. And normally this G is not close to the G24, but it's only in the distorted state that the G is brought close to the G24. And what happens when you mutate it to an A is that you form this additional bonding interaction with G44, then you form an extra hydrogen bond here. I mean, an AU is more favorable than a GU, and then in addition you have an additional interaction which only occurs in the transition state. Okay? And so that's why the HER suppressor, by lowering the energy of the transition state, again allows a read. So we've now explained a lot of the problems with the error rate. Now, how does the domain closure result in catalysis? Okay? So I told you that the domain closure results in EFTU, in the shoulder being pushed towards EFTU, and that actually results in a conformational change in EFTU itself. And the reason is that that loop of EFTU is stabilized by the shoulder, and in addition, there's a stacking interaction between the tRNA itself and the shoulder of the ribosome. And that stacking interaction also pulls the tRNA 3' end, so that this tRNA, which is normally next to a switch in EFTU that's very close to the active site. Actually, it's covering the active site and right next to the gamma phosphate. That interaction is being disrupted when you pull that away, and the reason that it's stabilized in the distorted form is an interaction with the shoulder of the 30%. So you can see moving the shoulder is resulting in changes in EFTU itself, so that the active site is actually being changed. Now, another thing that's being changed is the actual positioning of the EFTU. Okay? So that positioning is going to result in catalysis of the gamma phosphate. How does that happen? So I thought, since this is 2 o'clock, this is a 2 o'clock seminar, some of you may be dozing off. I thought this may wake you up. Okay? This is a guy who was a Bulgarian who defected to the West and became a journalist for the BBC. And you know this, what people hate are defectors because they really know the inside story, okay? And so this guy was a thorn in the Iron Curtain's side during the Cold War. And one day when he was standing at a bus stop in London, a guy came and stabbed him with an umbrella. And 4 days later, he died, okay? It turned out the guy who stabbed him was a spy from the Bulgarian embassy. And the reason he died was the umbrella was actually a little gun, which injected a pellet of ricin into him. And ricin is, you know, it's in the news these days because it's a bioterrorism problem. There's no antidote for ricin. It's a highly effective toxin. And the reason ricin works is because it cleaves, it depurinates a single base in 23S ribosomal RNA. If you ask where does it depurinate it, there's a highly conserved loop called the sarsen-ricin loop, where a conserved adenine is depurinated by ricin, and sarsen actually cleaves a highly conserved nucleotide. So it essentially cleaves the phosphodiester bond. So why is this sarsen-ricin loop so critical to ribosome function? Well, the first thing is, you notice the sarsen-ricin loop is right next to the GTP that requires to be hydrolyzed. And if you ask what's happening, here's what's happening. In the EFTU tRNA by itself, outside the ribosome, here's the gamma phosphate. There's a highly conserved histidine, which is positioned away from the gamma phosphate. And this switch 1 and P loop surround the gamma phosphate. So people thought that the switch 1, which becomes disordered in the GDP form, is acting as a hydrophobic gate, and is actually preventing a water molecule from coming into the pocket and resulting in hydrolysis of the gamma phosphate. Actually, it turns out that's not true. In fact, if people had looked accurately and looked at the structures and looked at the size of a water molecule, you'd realize there's plenty of room there for a water molecule. That's not what's happening. What's happening is the following. In the ribosome, this histidine is actually changing conformation. That's why it's really absolutely required for catalysis. And I mean, it's highly conserved. And it's changing conformation and is now coordinating the water molecule. And in fact, switch 1 doesn't get disordered. It's actually pretty much in the same conformation. And look, why is this histidine changing conformation? The reason is that it's being held in position by the sarsen-ricin loop. So all these induced conformational changes are such as to change the EFTU conformation such that the sarsen-ricin loop is positioned exactly correctly in the active site to coordinate the histidine so that it coordinates the water molecule. And then, so this is what happens. So you have the histidine in an inactive form outside the ribosome. Upon ribosome binding, you have the histidine which is activated, and then you have the activated water molecule that attacks the gamma phosphate. And after phosphate release, after GTP hydrolysis and phosphate release, this switch 1 then becomes disordered. And when switch 1 becomes disordered, EFTU changes its shape. Okay? And so that gives us a complete picture of decoding all the way from codon recognition to addition of the amino acid to the polypeptide chain. And that's as follows. EFTU comes in, it binds. If it's the correct tRNA, it starts sampling the codon. For that, the tRNA has to bend. Okay? But this bend conformation is an unfavorable conformation. And only if it's the correct tRNA is it stabilized by those three ribosomal bases that recognize the minor groove and give it additional binding energy to stabilize this bent form of the tRNA, which also results in conformational changes that position EFTU in just the right position for activation. And they also result in tRNA distortion and EFTU distortion. And then the sarsen rice group is positioned correctly for an attack on the gamma phosphate with the help of this conserved histidine. And when the gamma phosphate is hydrolyzed, the histidine moves away again, and then after the phosphate is released. So we have a whole sequence of events, and I've told all of that to you in words, but my postdoc and student, Rebecca and Martin, said that no one's going to remember anything that I say. In 10 minutes from now, you're going to forget exactly everything I told you, so they decided they would make a movie. Okay? And then they looked at the movie and they said, you know, you're going to just go blah, 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 blah throughout the movie, and they're not going to remember anything after the movie. So they said, it's better if we have songs for each of the steps of the movie, and if we choose the songs carefully so that the words represent what's happening. For instance, equilibration means, you know, the molecule is asking, should I stay or should I go? Minor group recognition could be something like Madonna singing into the groove. So if you have appropriate songs, then, you know, you'll remember the songs, and then you'll remember the sequence of events. Should I stay or should I go? Should I stay or should I go now?